Perfect. While we quickly transition for Dr. Eduardo Eizirik, my introduction for him, I finally will have the institution. He's coming from PUCRS, University in Brazil. I've also known him for over a decade, and I'm excited to hear about his work on jaguar populations. Everything is good, Stephen. Thank you, Stephen. You're awesome. And here's the advancer. And I think you're ready to roll. I don't think I forgot anything else.
Well, let's see.
Oh,
Is it?
yeah.
You guys hear me? Is it too loud? Is it too loud?
No. Good.
Okay. Okay, this works. This works. So, good afternoon, everybody. This has been a great workshop, so I'd like to congratulate first the organizers, Annabelle, Bridget, Chris, and Stephen, and thank them for inviting me to speak here. I was just noticing that I'm the last talk, apparently, and it's funny, is it? You think it is, right, for today? Yeah? So we started with tigers, and we're ending at least this block of conference with jaguars, so it's kind of a cool coincidence. So there are many similarities and some differences. This is something Uma and I always talk about. So Uma and I have been friends for a long time, so it's really fun to be in the same conference and to show some of the jaguar data and compare it to the tiger data. So as Bridget said, I'm at this university in southern Brazil. I'm also a member of several, and this is an NGO focused on carnivore conservation. These are two genomic and conservation consortia. I've been working on carnivores, ecology, conservation, and genetics for about 30 years. And we've worked on a variety of systems, big carnivores, small carnivores, other species that are the carnivore's prey. But today the focus will be jaguars. So I'm going to focus the whole talk on jaguars. This is a brief overview of a few points that you guys can go through while I'm talking. I'll highlight them as I go. This is the light gray, the original range of jaguars in historical times, about 1900 till today. So it used to range from the southwestern US to mid-Argentina, and then it's been wiped out from over half of its original range. This is dark gray is an optimistic view of its current range. It's actually much worse than that. So it's lost over half of its range, over half of its individuals. There are a few strongholds, such as the Amazon forest here and the Pantanal here, and the Mesoamerican rainforest in the Yucatan Peninsula. But the majority of the populations now are small and isolated. Some of them have 5 individuals, 10 individuals, 20 individuals. So it's really similar to what Uma described for tigers in India. There are many issues in terms of habitat fragmentation, hunting, prey depletion, human-wildlife conflict, and we've been working on their genetics for a while. This is the first study we did in 2001, which looked at mitochondrial DNA and microsatellites across the species range. We showed that the species has pretty good levels of genetic diversity compared to other large predators, and there's quite compelling evidence for historical range-wide connectivity. So there are some disruptions, some putative historical barriers, such as the Amazon River, but they are subtle, and you actually don't see it in all the markers. So mitochondrial DNA, for example, shows some evidence of disrupting historical gene flow because of female philopatry, but male biased gene flow probably compensates for that. In the nuclear markers, we generally don't see this structure. And this idea, this inference that we put forth over 20 years ago has been the basis for jaguar conservation strategies globally. So now the NGOs and the governments, as they plan for jaguar recovery and jaguar conservation, they take into account this idea that there was historical high connectivity across the range. A particularly concerning part of the range, I'll just move back one, is this area here. This area is the Atlantic forest, so it's a tropical biome on the eastern side of South America, and the situation of jaguars there is particularly critical. So this is a letter we wrote 10 years ago highlighting the plight of jaguars in the Atlantic forest. It may become the first tropical biome in the world to lose its top predator. There are less than 250 individuals left in the Atlantic forest, and they're all partitioned in these small populations. We've been studying their genetics for a long time. And before I go into some of the genetic studies, I'll just show you a map. The original range of the Atlantic forest here, you see South America there. It used to occupy about 2 million square kilometers. It was a large biome. And most of the Brazilian colonization by European settlers started in the Atlantic. And so a lot of the deforestation, a lot of the urban development, agricultural development has happened exactly where the Atlantic forest was. So now we have less than 10% of the Atlantic forest. And here on the right, you see in gray where the Atlantic forest was. And these are patches where suitable jaguar habitat remains in the lighter shade, and where jaguars actually still occur in the darker shade. So I'm going to talk about some of these fragments. The first study we did came out in 2010. It was a study using microsatellites in which we looked at this portion here called the Inner Atlantic Forest. So this is a blow-up of that. This is the largest block of Atlantic forest in Brazil. These are Iguaçu. This is Iguaçu National Park, where you have the well-known Iguaçu Falls. They're here. So you still have a population of jaguars here, and then in Argentina here. So this was the first time that we had reasonable numbers of individuals here to look at population level processes in jaguars. A sad part to it was that the first population in the world to be able to be sampled was this one, which is a population that occurs here. And by the time we published the study, the population was extinct. So the only place where they were still hanging in there was around this river, on the edges of the river, and this was a hydroelectric dam that was flooded and then destroyed the remaining habitat. So we lost the population before we published the first study. So what did we find? This is a structure plot with microsats. Each of these areas here is a location, one of those patches. And this was highly remarkable. You see much more differentiation across these different patches than you would expect. So each of them has its own genetic cluster. A few individuals that are migrants. So this was caught in the yellow area but came from the green one. This was in the blue but came from the yellow. And this is an admixed individual. So first, the patches were highly differentiated, but there was evidence of movement and even gene flow between them in 1998 to 2002. This is where these individuals come from. So they are from the late 90s, before the flooding of that dam. And it looks like all the connection has been severed after that. This is a map of reconstruction of how the forest cover looked like in 1900, 1940, 1970, and in the late 90s. So this is the northern block, this is a southern block, that's where that dam is. This is a bigger dam, Itaipu Dam, that severed the connection between the southern and the northern block. So it looks like in just 30 to 40 years, or 6 to 8 jaguar generations, drift has led to the differentiation of these populations to an amazing extent. It's quite surprising because jaguars can easily move from this part to that part in a day. So they can walk 20 to 30 kilometers in one day, but it's remarkable, and radiotelemetry studies corroborate that they don't. Basically, they don't go across the matrix. When they do, they die generally in the process because they prey on cattle or they're just killed upon sight. So they don't make it across from one patch to the other. And when they do, eventually, they tend not to breed because they're already at carrying capacity. There are like 8 jaguars in there, and that's all that that fragment can support. So this was something we published at that time. We had a problem with reviewers because they actually didn't believe us. They didn't think that it was possible that in just 30 to 40 years of isolation we would get such a remarkable differentiation among patches. And so we started since then to accumulate data from other species, which I'm not going to show, but we've been doing similar studies for other carnivores, for other species, just to show that they don't have this pattern, and that's what we see. Other species don't show this pattern of differentiation among fragments, and we have been looking at other areas, jaguars in more continuous habitats. For example, well, before I go to the Pantanal, which is the next one, this is another patch. I just included another patch here, which is even more isolated. The one we were looking at was here. This is in the coastal Atlantic forest. It has a very small size with both camera trapping and non-invasive DNA identification of individuals. We got about 10 to 11 individuals census size. Effective size we got was 8 individuals. This is a structure plot with microsats using different K values, and the green is this patch here. So again, we see a pattern of low diversity, strong differentiation relative to other patches. Now we go to the Pantanal, which is here. It's a large floodplain. It's a large jaguar population. As far as we know, it's not fragmented. So initially we looked at these southern populations, and these are just other structure plots with different K values. And here was like a test case in which there is no fragmentation. The distance between these areas here and the areas we had been looking at in the Atlantic forest is not that different. So we're using it as a control. And we see the Pantanal here, mostly unstructured. We did several tests and it seems to behave as a single population. Then we moved on to the northern Pantanal. So there's a sampling gap in between, which is a problem, but most people doing field work are either in the south or in the north. But it does seem to be still a mostly continuous landscape. So with this study, we saw some structure. You see a trend for structuring south to north. But if you look at a PCA or other multidimensional scaling analyses, other multivariate analyses, or an isolation by distance plot, geographic and genetic distance plot, you see most an isolation by distance scenario in which this population here is almost half and half between the two genetic clusters. We think the Paraguay River here has been a major avenue for dispersal. There are many studies with landscape genetics going on, and we see that there is not suitable habitat in this area really, so there are fewer jaguars. Not only there are few people studying jaguars, but there are fewer jaguars here. So they're actually right in focusing their studies in the northern and southern areas, but we'd like to see more sampling going on, at least on the western end, so that we could test this hypothesis of gene flow over through the river. Another interesting thing we did in this same study, came out a couple years ago, was the first pedigree that you could estimate for jaguars in the wild. Because there are many molecular ecology questions that we'd like to address, there are behavioral questions, but also for the genetic studies, population genetic studies, evolutionary studies that we do at a deeper scale. We don't have, for example, generation times estimated for jaguars, and this was the first time we estimated one based on pedigree. So this was a population in the south, this population here, for which there were people in the field for 22 years. So they have actually captured jaguars and sampled jaguars, and then we can reconstruct their genealogical relationships in the south. And these are, the colors are the different sampling sites, and you can see that the pedigree spans the different sampling sites. They move across the landscape readily, and the similar pattern is emerging from the north as well. So that's an interesting development. We have been using it in other studies as well. We then included the Amazon. So this is a study by Gustavo Lorenzana, PhD student in the lab. Our largest microset data set with 109 individuals. And you can see the Amazon here. This is a bar plot, and this is just a landscape plot of the same data. You see the structure here in the Atlantic forest within that highly fragmented area. The isolated patch here actually has a similar allele composition as the southern block here, which to us indicates that they still harbor some original alleles of the Atlantic forest. What I'm going to highlight here is the Amazon. We have a 3,000 kilometer transect across the Amazon and no evidence of structure there, not even the Amazon River. So to us it gets stronger and stronger, the inference that there was historical connectivity across the biomes, although we see some differentiation among these biomes. We started moving towards genomics in 2011. We sequenced the jaguar genome in 2014, and the paper came out in 2014. So this was Enrique's work, who's right there. I'm going to take a picture of your slide. And we did a lot of different analysis. It was mostly phylogenomics and introgression analysis at deeper temporal scales, which I'm not going to go into today. It was a very close collaboration with Bill Murphy at Texas A&M and his postdoc Gang Li and several other collaborators. And what I'm going to just mention from this study is that this included the generation of an exome probe set for about 19,000 genes. And we started using this exome probe set to perform population level studies as well. So one of these studies was also performed by Enrique. And I'm just going to show one slide from it. We have the different biomes, 109 individuals, 30,000 SNPs after filtering. And so we're looking at biome-wide differentiation. So we're looking at genes that are indicative of adaptive differentiation among biomes, and we find several. So it's interesting that we see in jaguars something similar to humans. I mean, they have large-scale connectivity, they have no barriers to admixture, but they do have regional differentiation, and that's what we're trying to understand and use that to enable conservation programs, which is where I'm going to get at the end. So you see genetic clusters, you see lot of sharing of clusters across biomes, but you see clusters that are predominant in one biome or another. So that's part of our assessment for recommending how much we should mix jaguars from different biomes in our ex situ programs in Brazil, for example. So this is a partnership with the Ministry of the Environment, and they want our advice on how much to treat jaguars as a single thing or each biome as a single thing. Obviously, we cannot treat each local population as a single thing because they're minuscule. And fortunately, we don't have to because they're clearly derived from human-induced separation. So we started moving as well into whole genome sequencing. This paper that came out last year was also part of Gustavo's PhD project. It included 13 complete genomes from jaguars, mostly from the Brazilian biome. So you can see the biomes here. One from Guatemala and one from Arizona. It's the only jaguar that has been caught and sampled in the United States. And so we're fortunate enough to get that genome and be able to compare it with the genomes we have been analyzing in South America. So I'm going to go through some of the results from that paper. This is a PSMC plot, and it's been really interesting to see the discussions here about PSMC. And here, regardless of whether we interpret it as population size fluctuations or connectivity, I think the inference is the same, or both. It's quite remarkable that jaguars from all these biomes, including Guatemala and Arizona, they have very concordant trajectories here, which is highly suggestive of the same demographic history. They were connected throughout this time, which agrees well with our previous inference. So they have a decrease, have an expansion, they go down, and the only thing that is different are the two Central American and North American individuals. They seem to have had a little sharper decline, and it's still tens of thousands of years ago, not anthropogenic. We know PSMC is not sensitive to very recent demographic processes. We interpret this as a consequence of sequential bottlenecks as they colonize the northern edge of the range. So it's been expanding from south to north. We've been getting that signal in other analyses as well. And as they reach the far northern end of the range, we see a signature of sequential bottlenecks. If we take a look at heterozygosity, so these are jaguars from the different biomes, and in this case we compare to lion and tigers from the different regional populations or subspecies. You see this is SNV rate, so the proportion of SNVs, single nucleotide variants per window of genomic sequence. Each individual is a black dot. Atlantic forest has a lot of variants. The average per biome is a colored dot. But you see, jaguars are mostly variable, more variable than African lions. This is the Asiatic lion that Uma mentioned, so it has very low variation. These are tigers from different regions, and mostly jaguars are quite variable, and except for the Guatemala and Arizona one, which have decreased variation within the range of tigers. If we look at ROH load, am I in front of you? Can you see?
No, you're good.
I'm kind of, because I tend to walk around. I'll walk this way. So if you see the sum of ROH and sum of size, of length, and the number of ROH, you see a pretty interpretable plot. You see the Arizona one here at the top with the most runs of homozygosity, which agrees with our inference of sequential bottlenecks as that area got colonized. The Guatemalan one is here. And you see quite low burden of runs of homozygosity in some of the other animals. And it's quite interesting to take a look at these two, which come from those same patches that I had shown you earlier with the microsatellite data that are isolated patches of Atlantic forest. You see an excess of long runs, which is implied by this deviation towards the right in the sum length relative to their number. So this is an interesting result. We've been exploring this some more, as I'm going to show you in a second. The latest round of this analysis is based on a larger number of whole genomes. We now have 61 whole genomes from jaguars sampled mostly in Brazil. There are those two from Guatemala and Arizona, which I'm not going to show here, so it's 59 if you don't count those two. We're trying to get a pretty even sampling across Brazil. This is the work of Karen Sartor, who was a postdoc in the lab, and Gabrielle, who's a graduate student, and Enrique has been involved as well. So these are the samples we have. These are the biomes. For the first time, we have a sample of this red here. This is the Caatinga biome. In the previous study, we had one animal. Now we have all the animals that have been caught in this biome, which are 5. So there are only 5 captures in the whole biome. This is a dry, scrubland-like environment, and jaguars are pretty different there ecologically and behaviorally. So we're interested in adaptation as well as assessment of load and assessment of inbreeding in the 2 remaining populations in this biome. And you know there are these patches Should I continue? Right?
Yeah.
I'll just continue.
If you turn that off, we don't have any feedback.
I think it died.
Yeah.
All right. So we're interested particularly in those patches and comparing them to large populations such as the Amazon here and the Pantanal there. So if we first take a look at a PCA plot, comparing those biomes, the first thing that strikes us are these guys. They're real outliers, and they come from that coastal isolated patch from the Atlantic forest. They are the most different ones in the PCA. If you take a look at another set, these are two different patches, but the two individuals still clustered together, which is indicative that they haven't been isolated for very long. So it's a short-term isolation, a few decades probably, but still enough to drive them to low diversity, as you're going to see. The Caatinga ones, two of them also form an outlier cluster. So we're seeing the effect of drift in over a few decades, or several decades perhaps in this case, decreasing diversity, as we're going to see, and increasing differentiation. Here you have mean heterozygosity. I'm just going to highlight those two patches. So each individual is here. Generally, jaguars have high levels of heterozygosity, as we saw in the previous slides, but those three individuals from that area and the two individuals from this area have particularly low levels of heterozygosity genome-wide. If you look at the FROH, which we have been seeing here at this conference, same thing, quite a lot of burden. Here we have almost 40%. Here it's over 40% of the genome in runs of homozygosity in this individual here, which is one of just this individual there. And this other one is from this patch. So actually the record holders right now are from that patch with about 40% of their genomes in runs of homozygosity. Most of the others are doing pretty well, but our interest is in contrasting patterns of diversity, runs of homozygosity, what's inside those runs, not only to understand the evolutionary implications, but also to make recommendations for conservation. This is an SNV plot that you saw with both Enrique and Klaus showing. We can see this is from one chromosome, one of our sampled chromosomes across individuals. You can see the runs, or at least the areas of low heterozygosity that are quite coincident with runs of homozygosity. They vary across individuals. This is one chromosome, this is another chromosome. I just brought three, but you can see how the runs vary across individuals, and we can actually map them per biome, per fragment. For example, here I'm going to mask the others and just look at the Atlantic forest ones. So just mask the others, and these ones here are from the Atlantic forest, which are individuals that are likely going to be used for reproduction in captivity, or for reintroduction efforts or augmentation efforts, or other individuals that are still unsampled that we might have the ability to collect or to put in captivity. That's a discussion about what to do, how to actually promote connectivity, to restore connectivity. But if we just take a look at, for example, the top individual here, this run is shared by two others and partly by others here. Here we see shared runs as well. So we're in the process of comparing runs, looking at shared runs within a patch, among patches, and thinking of how we can use this in terms of making recommendations for conservation. For example, here, I'm just getting the three individuals from that worst patch, this patch that's here. So just looking at those three, you see how much shared homozygosity we have. One thing that we're interested in looking at is not only whether the segment is shared, but the haplotype, because they might be fixed for different haplotypes, which in the end would be a good thing if we mix them either ex situ or we promote their admixture through connectivity in the wild. That would be a useful thing to know. So that's what we're doing at the time. So the next steps here are to improve our estimates of NE based on whole genome data, per population, per biome, assessment of genetic load using tools such as the ones you have been talking about here at the conference, address the relationship between ROH and load, so look at the deleterious variants and their positions inside or not inside the runs of homozygosity, and what I just mentioned in terms of haplotype differences among runs. Things that we're thinking about, I don't know how much time I have, am I
7 minutes.
I have 7, so that's going to be good. You guys can actually ask specific questions and we can discuss some of these potential management actions. Might be a good thing to have a go at management alternatives for situations such as these jaguars. So one thing we're doing is to use this data to make breeding recommendations for ex situ managers. So this work is partly funded by the Zoo Association in Brazil. So there's a jaguar breeding program, and they want to know which individuals to breed, which biomes to prioritize. Obviously, we know that the Atlantic forest is a biome that needs to prioritize, but we know very little about the Caatinga. So they're eager to hear about how different the Caatinga is, how much it has to be kept separate. That's an ongoing discussion, how much we need to keep the biomes separate or mix them, and hopefully we will be able to aid them using these genomic data. Then harder than that, to some extent, is what to do with these isolated populations. I haven't really told you much about this one, for example. This is a population with extensive camera trapping efforts and whose census size is 9. So there are 9 jaguars in that area, and there have been 9 jaguars for a while. There was an individual that got caught about 300 kilometers south of it in a botanical garden. Nobody knows where it came from, so they caught it and decided to release it there. So there has been a successful reintroduction or translocation of an individual, and they have been following it for 4 years, and apparently it has bred. There's evidence of cubs in the area where this animal is occurring. So we haven't received a sample of it yet. And there's some controversy as to should they have translocated the animal or not. We haven't sequenced its genome yet, but they have collected the sample. But that's one of the few successful translocation efforts with jaguars. They are hard to translocate. They have behavioral issues with staying where they are released. They tend to try to go back to their original site, and they usually die in the process. So translocation is complicated. These 2 patches, for example, are 400 or 500 kilometers apart, with lots of industry, agriculture, cattle ranching in between. So it's really not feasible to reconstitute corridors here, much less here, much less there. Intensive interventions such as reproductive technology applications, such as artificial insemination, in vitro fertilization, embryo transfer, these technological interventions are being considered as potential tools. They are hard to implement, but corridors are impossible in some of these cases. Translocations are a possibility, but they are also difficult. And there's an interesting avenue that people are considering, which is to have semi-captivity of local individuals with the transport of an individual from another patch, for example, a female from captivity, and leave her in semi-captivity until she's in estrus and she's in heat. And then a male could naturally come into the enclosure. So there are ways of letting a male into the enclosure without letting them get out of the enclosure, and wait until they breed, and then the male goes back out and they keep the female ex situ or take her back to her patch, hoping that no other female has taken her territory by then. But this is so far being considered mostly for ex situ females. So inseminating ex situ females with males from other patches. So that's the kind of ideas that we have been debating, and we can talk more about it if we have time. I'd like to thank my colleagues and collaborators, students, postdocs who have helped in these jaguar studies over the years. These are the funding organizations that have helped us perform these works, and institutions which have also helped. Thank you very much.
Yes, you will do. Questions, hopefully students and postdocs first, then we'll move to others.
I'll take this out because it's not working.
You first.
Yeah.
Oh, yeah.
Hi, really cool talk, super interesting. I just had a question on the like SNP density map. I noticed so like most individuals had sort of a few long sequences of low variance, and then there was like one individual, BPON 302.
Which one? Well, all of them.
Yeah, BPON 302.
Yeah, this is a
whole bunch of short.
Yeah, this one we're pretty sure it's an artifact of low coverage, so we need to remove this animal.
Okay.
But there are others, and it's there for all the chromosomes. So yeah, we did not get enough coverage for that animal yet. So I think that's probably the reason. But yeah, if you look at the other ones, even animals from the Amazon, for example, where's 404, this one, it's from the middle of the Amazon, and it has a large tract there. So it's kind of a conundrum. I mean, it's like the most pristine area of the Amazon. But maybe there are behavioral aspects. There are people in the field doing camera trapping and radio collaring of those jaguars, and they seem to stay there. This is an area that gets flooded annually, and they live on the trees. They actually don't. People thought they would get out during the flooding season, but instead they actually go up in the trees. They have cubs. They stay up in the trees. So maybe they have behavioral issues that keep them there, and they don't breed much with animals from elsewhere, and maybe that has to do with some inbreeding that's behavioral induced. So these are other layers that we are not even aware of, but it's possible that they are affecting this type of pattern in addition to anthropogenic processes.
Super cool talk. I was I was just curious about when you showed us the plots of estimated population size. It dips and then recovers for all of the different jaguar populations. And so do you have any thoughts about whether there was anything happening climate change wise or anything around that time when it dips and then recovers?
Yeah, so that's always a good question. And there are two situations when you consider these questions. When you are the author of the paper, you come up with an explanation. When you're a reviewer, you ask that question. So you say, do you have an explanation for that? So everybody has hypotheses of what was going on. So first we heard here that it could be both size or connectivity, or both at the same time. We could guess, both in jaguars and in other species we've been working with, that it is glacial cycle related, because it does match glacial cycles generally. So you see a dip and a recovery, and there have been 18 glacial cycles since the beginning of the Pleistocene. There's variance around the estimates of the cycles, and there's variance here. So we can always argue that it has some connection with the glacial cycles, and probably does. But yeah, that's my current hypothesis, that glacial cycles are affecting availability of prey, availability of habitat, and that is a driver for demographic cycles. And we see that in pretty much all the carnivores. We've done otters worldwide, and we see actually concordant patterns. We've done it with canids, and so with other cats, and they all have cyclical ups and downs in terms of effective size estimated by PSMC, which sometimes agree, which is kind of neat when they actually have the same pattern. One pattern we saw was opposing cycles. We have a grassland species and a forest species that had exactly opposing cycles. So that gave me more confidence that it is glacial induced, because when there's a glacial cycle, grasslands expand and forests decrease, and when you have an interglacial, it's the opposite. And that's exactly what we saw in those two small cats of South America. So in that case, and the otters, I'm confident that it's glacial related. Here, I would guess it is as well.
Very cool.
Thanks. Nice talk. I was curious about ecological differences maybe between these, because they're very different habitats, right? Mm-hmm. So are there different diets? Do you know anything about whether these animals they eat different things, or ecologically are they different in any way? Maybe the prey density is also not able to support higher densities because of the habitat or something like that.
Right. So clearly here, this is the case. This is very dry forest. This is really peripheral habitat. Densities are low, prey densities are low, so for sure, there's not a lot of prey availability here. Mm-hmm. In the Amazon, in the Atlantic forest prior to human intervention, in Brazilian biomes in general, prey availability is assumed to have been pretty good. But there are differences, and the most striking difference is in the Pantanal, this area here, where jaguars are notoriously able to hunt caiman, which are large alligators like. And I mean, if you are interested, you can go to YouTube and see jaguars diving from banks of rivers and coming out with a huge caiman in their mouth. And there's actually very interesting evolutionary questions that we're trying to address. In this paper, in our genome paper, that's one of the things we saw. We saw signatures of positive selection in genes involved in craniofacial development and forelimb development. And we connected it to a hypothesis that was put forth by Louise Ammons, a famous mammalogist from the Smithsonian in 1987, which postulates that jaguars are the only panthera cat that survived despite the megafaunal extinction by switching to armored reptiles. So jaguars do hunt a lot of caiman and turtles. They have the strongest bite among all the cats, and they can break through the shell of a turtle. And so they eat turtle and caiman regularly, at least in some areas, and we saw signatures of positive selection in genes related to that kind of developmental feature. And the animal we used was a Pantanal jaguar. So one of the things we haven't done yet is to go back to those genes and see if all the individuals have those signatures or only the Pantanal one. But clearly, there are differences in diet, there are differences in prey availability, but it looks like jaguars are quite flexible. So although there probably will be local adaptations or local behavioral features, they seem to do well and go across, like for instance, this whole area here in the middle is a savanna-like environment that has signatures of connectivity both towards the north in the Amazon, towards the east in the Atlantic forest, towards the Pantanal. So the connectivity seems to have been a feature with local adaptation at the same time.
And just one other quick question. I seem to remember something about either WCS or Panthera having a large program for corridors.
Yeah.
So what is that just talk, or is there something really happening on the ground, and can you look for the effects, potential beneficial effects of that for connectivity?
Yeah, so it was Panthera. I think it started with WCS and then Panthera. And one time I was talking to Alan Rabinowitz, who was the leader of that program, and he told me that was because of the paper, but because of the 2001 paper. And I was like, well, really? That's cool. It's rare that you see a genetics paper used so directly to, enable a conservation strategy. But yeah, since then, I mean, they decided to do it a few years later, but they have been promoting connectivity. How is that done? Well, it's done nationally. So it's hard to promote range-wide connectivity. There's several gaps by now, but they're working with local governments and they're creating national parks. I mean, I don't know. I don't think it's just talk, but I think it's hard to actually make it work, in the face of deforestation, in the face of development. There are many gaps that are, in private lands. They need to make them into parks. So yeah, it's difficult to actually get it to work completely, but they're trying and they have had some successes.
I think we'll take one more question and then we'll break.
Yeah. You just answered the question because it was about local adaptations, if you're going to look into that or if you have looked into that, because you have a wide range of environments that you could look and see if there's local adaptations, but you just answered that.
Yes, so we are looking into that, but it's actually an interesting challenge to look at adaptation in the face of so much drift, right? Maybe the Amazon and Pantanal will be good systems for that because they are larger populations that are ecologically distinct and that have had less of an anthropogenic effect of drift, whereas the Atlantic forest is quite important in terms of management, but harder to study because each patch is quite different from each other due to anthropogenic drift. So we're still interested in looking for adaptation there, but I think it's going to be a little harder.
So we have more time later for you to go find all of our speakers except Diego and ask them more questions. So let's thank Eduardo for his talk.